Everybody, this lecture is going to be focusing on the cell organelles that you would find inside of a eukaryotic cell. We'll be going through all the different functions and give you some analogies of each one as we can. First of all, with the cytoskeleton. Like the name would guess, remember the prefix cyto refers to cell. So this is the supporting framework of the cell. There's three kinds of filaments you need to be aware of in this class. The microfilaments, which are basically made out of actin and are really essential in the contracting and moving around of particles. They're really common inside of muscle cells, where they're especially modified to provide the muscle cells to contract, so we can move the food through the digestive system or work against bones here to move joints. You also have microtubules. These are the largest strands, and like you would guess, tubules are made out of a protein called tubulin, which makes a tube. Inside the cell, these are really common with the organelles called the centrioles, which are responsible for moving around chromosomes during cell division. Now you got the smallest, which are the microfilaments, and the largest, which are the microtubules. In between, we have the intermediate fibers, which main function here is to anchor the organelles in place. So summing things up here, microfilaments, microtubules, their main function here is to function in cell support and movement. Holding the cell together, making parts move. Now you will see inside of a eukaryotic cell is the nucleus. The nucleus in this picture here, shown right in the center. And in our actual picture right here, here's the cell membrane. And this purple area here is the nucleus. The nucleus, unless you look through an electron micrograph, you see that it's covered with a nuclear envelope. So that's why this diagram down here is kind of important. You look at the nuclear envelope right here, okay, double-layered membrane, and periodically throughout here you have little tiny pores. The pores are there to allow products from the nucleus that are made inside the nucleus to leave. When we talk about protein synthesis, in this picture right here you see a particle called messenger RNA leaving through the pore. Okay, more about that when we start talking about how proteins are actually made. But also inside here, you're going to have the DNA, which has to stay intact. The DNA, too large to fit through those pores, so it stays within the nucleus. Also inside the nucleus, you're going to have a small nucleus called the nucleolus. Yeah, they sound a lot alike, and the spelling looks a lot alike. But nucleolus literally means little nucleus. In our pictures here, if you look at this diagram here of a nucleus, within the center you've got the nucleolus. And here's the nucleolus in a real cell, and all the DNA scattered around through the outside. The job of the nucleolus is to make ribosomes. Ribosomes, which are going to function in the cytoplasm of the cell, are responsible for making proteins. I just mentioned ribosomes, probably should start with them. Ribosomes are found in both prokaryotic and eukaryotic cells. Now remember, a prokaryotic cell lacks membrane-bound organelles. Well, no problem, because the ribosomes have no membrane. Ribosomes inside a eukaryotic cell, however, may be either free or attached. By free, I mean simply floating around inside the cytoplasm. By attached, I mean, like in my picture over here to the right, they're attached to a membrane. This membrane here is the endoplasmic reticulum, which we'll look at on the next slide. But you look at these little tiny dots right along here. Those are the individual ribosomes. Now, a ribosome made inside the nucleolus is composed of a nucleic acid called RNA and proteins. Its function is to make proteins. Therefore, a common nickname for the ribosome is to simply call it the protein factory of the cell. Now, I mentioned the endoplasmic reticulum as this membrane right here that has the ribosomes attached. We'll go on to that one next. The endoplasmic reticulum is a membrane system that extends from the nucleus out to the cell membrane. Basically, I put up there, it's a pathway to move materials in the cell for them to move around. I would suggest that you maybe think of the endoplasmic reticulum as a highway system or a railway system, where materials are packaged up in one spot and moved to the other. Now, in a cell, there are two kinds of endoplasmic reticulum. There's both smooth and rough. The only difference is this. Smooth ER, not covered with ribosomes. So instead of they being riddled with ribosomes, and here's a diagram of what you saw on the previous slide, instead of having the ribosomes all attached here and giving it a rough looking appearance, like sandpaper, they're smooth. Now, smooth ER is responsible for breaking down poisons that enter the cell, okay, as well as also making lipids. Now, the rough ER gets its name because it looks like sandpaper. It is covered with those little tiny ribosomes. Now, the proteins that are made here are going to be responsible for being exported from the cell. So if we've got a cell, for example, of the pancreas, which is making pancreatic enzymes here for digestion of food, you know, those proteins are going to be made on the ribosomes that are attached to the endoplasmic reticulum, so rough ER. Associated with the endoplasmic reticulum is something called the Golgi apparatus. This is an organelle that goes by several names, Golgi apparatus or Golgi body, you know, or Golgi complex. You know, all three of them I've seen used in different textbooks. The job of the Golgi apparatus is to package up the proteins. So proteins that are going to be made by the rough ER and transferred along through the endoplasmic reticulum find their way to vesicles. So as I put up here, what's going to happen is a vesicle from the rough ER is going to be produced, and it's going to come towards the Golgi complex. Now, once it's embedded inside the Golgi complex, it passes through the Golgi complex, or Golgi apparatus, if you will, and all along this way here, those proteins, shown right here, are being modified. They may be trimmed up a little bit. They may have sugars or other particles attached to them. Basically, they're modified and packaged up. So the main function here is to get the proteins ready for export. So if you think about this like a factory, the items in the factory are made on the production floor. They are then sent to the shipping department where the products are being packaged up into bundles of 12 or 24 or whatever they're going to sell them as, labeled up with a price, for example, and shipped off with an address label off to a certain destination. And in this case, those proteins, they're passed along here, pass off into vesicles, which go towards the plasma membrane and are then released. Many eukaryotic cells are lysosomes, which have been nicknamed the suicide sacs of the cell, because technically, if they were to release all their digestive enzymes at once inside the cell would be destroyed from the inside out. But its main job would be to break down food. So in one-celled organisms, for example, that maybe eat a bacteria, you know, they'll form a food vacuole, and the food vacuole then will pair up with a lysosome, kind of like you see in the picture over here, that has all these digestive enzymes. Once they merge, the digestive enzymes break down the food, or as shown in this picture here, an old worn out organelle is being digested, and the materials from the broken down organelle are then basically going to be returned to the cytosol, so they can be used to make new organelles or to recycle those parts altogether. So basically this is kind of like a recycling center. You might want to think of the lysosome like a junkyard, where old cars are taken, dismantled, and the parts that are still functional are passed on to, you know, other people going to repair their vehicles. But lysosomes also play a role in development. You know, late in embryonic development, you know, we have webbing between our fingers. Well, very seldom are humans born with webbing between their fingers, because the lysosomes in that tissue are programmed, hardwired, I guess you might say, to release all their lysosomes at once and destroy that tissue. So you don't see, you know, humans being born with webbing between their fingers or toes. I briefly mentioned a food vacuole. That's one of the three kinds of vacuoles you'll be responsible for in this class. The other two are the contractile vacuole and a central vacuole. Contractile vacuoles, we'll start there, are found mostly in one-celled organisms, like an amoeba, a paramecium. What happens is they work like a sump pump in a house. Sump pumps work by collecting water around the basement walls, collect them into one spot, and the sump pump then pumps out the excess water. Well, the contractile vacuole takes up all the excess water out of the inside of these one-celled organisms and pumps it out. This prevents the cell from exploding. So you're going to see these contractile vacuoles, especially in freshwater one-celled organisms. And in the next chapter, we're going to look at why the water actually rushes in. In plants, you'll find a central vacuole, where ions, nutrients, basically the storage materials of the cell are going to be maintained. Okay, that's what we're showing over here. Your typical plant cell has this huge central vacuole where a lot of these materials can be stored. Now, in one-celled organisms like amoebas, paramecium, and things like that, when they engulf a food particle, it's closed off into a vacuole. Therefore, it's called a food vacuole. Now, remember, that food vacuole then is carried towards a lysosome, where it binds with the lysosome, and the digestive enzymes from the lysosome break down the contents of the food vacuole into smaller parts, used for nourishment or for building new cell parts. Organelles, we're going to spend a whole lot of time with. Matter of fact, in our textbook, we'll spend a whole section of the chapter here devoted to each one of these. We're going to start with the chloroplast. Now, the chloroplast's job, and the chloroplast is found only in plant cells, is to make sugars. So therefore, I've listed here as a carbohydrate factory. The process of making sugar from water and carbon dioxide is called photosynthesis. So here, your typical chloroplast right here, both in a drawing and in an actual electron micrograph here, shows what they look like. In essence here, it is a double membrane. So you have an inner and outer membrane. Now the outer membrane is very, very smooth, but the inner membrane is folded up into these flattened sacs. Now when we actually study the process of photosynthesis, we're going to look at these sacs very, very closely, and we'll start identifying the parts of those in that chapter. But in the meantime, just remember, this is where photosynthesis is occurring, and photosynthesis is the process of making carbohydrates, in particular glucose. The last structure here is the mitochondria. Mitochondria are found in both plant and animal cells. Many students kind of get the impression that photosynthesis, carbohydrate or chloroplasts are found in plants and they make sugars. Well, the mitochondria's job is to break down sugars. So a lot of students believe that they are found only in animals, but in reality they are found in both plants and animals. The mitochondria's job, like I said earlier, is to break down sugars and release power. So we supply energy. So this has been nicknamed the powerhouse of the cell. And like the chloroplast, mitochondria is surrounded by two membranes. Again, the outer membrane is very, very smooth, and the inner membrane is folded up into little compartments. So as you're looking at these right here, here's your smooth outer covering of the mitochondria, and here are the many folds going on the inside. So this inner membrane going right along here gets folded up many, many times. This increases the surface area because on this membrane that we have, the process is going to produce the most energy in the form of a molecule called adenosine triphosphate. We abbreviate that ATP. Now, these little folds have a particular name. They're called cristae. It's kind of like the girl's name Christy with a long A at the end. But those are the individual folds on the inside of the mitochondria. Again, all to increase the surface area of the cell.